I'm Jeremy Kaplan. In this video, I'll be discussing abortive transcription, focusing on figure 6.7 and 6.8 from Robert Weaver's 5th edition of Molecular Biology. This video was made for MCDB 427, Molecular Biology at the University of Michigan. In this video, I'll be discussing abortive transcription and one of the first experiments that showed that it could happen. In order to understand what this means, we first need a brief overview of the process of transcription initiation in prokaryotes. Prokaryotic transcription begins with RNA polymerase binding to the promoter region in the closed double helix form. The sigma factor of RNA polymerase then stimulates the polymerase to open the double helix, exposing the single-stranded template. The polymerase can then incorporate a few nucleotides into a growing chain. Here's where abortive conditions matter. DNA-RNA interactions are typically quite strong in larger numbers. However, short hybrids can be quite unstable. At times, the newly synthesized RNA can dissociate, aborting the transcription process. This results in an abortive transcript. The polymerase can then try to synthesize the transcript from the beginning, and will repeat trying to do so until it disassociates or fully elongates the transcript. Once elongation begins, the polymerase can clear the promoter and sigma factor can disassociate. Here's a great example of how those abortive transcripts can appear. Researchers set up two different in vitro transcription reactions with the T7A1 and T5N25 promoters. RNA products were N-labeled with gamma-labeled 32P-ATP, and the reactions were incubated with heparin so that each RNA polymerase could not reassociate with a DNA molecule after disassociated at the end of transcription. The researchers used two different promoters to show that the rate of abortive transcription can differ based on the promoter strength. You can see in this data that each reaction resulted in quite discrete band populations. This large band, labeled plus 50, was the full-length transcript, and these discrete bands down here are the abortive transcripts. You can see that we get band sizes from 2 to 10 nucleotides long, but get no finished product longer than 10 nucleotides until the full transcript. This is pretty clear evidence of aborted transcripts, but one of the first experiments showing this, figure 6.7, wasn't that simple. Let's first talk about the experimental setup for figure 6.7. The researchers used a DNA construct with the lac UV5 promoter, whose transcript on the non-template strand began with this sequence. The researchers incubated this DNA construct with RNA polymerase. After 10 minutes, heparin was added. Heparin is a negatively charged polysaccharide that prevents RNA polymerase from binding to DNA. By adding heparin after RNA polymerase, the researchers ensured that RNA polymerase would not bind a new DNA molecule after falling off the template. It will also help to show later on that after abortive transcription, RNA polymerase stays on the DNA. Otherwise, full-length transcription would never occur. The researchers then added various concentrations of 32P labeled ATP and unlabeled CTP, GTP, and UTP. Each reaction was stopped with EDTA and then ran in a separate lane on a polyacrylamide gel for maximum resolution. The gel was then exposed onto X-ray film to identify the sites of radioactivity. Let's first get an understanding of these results at a broad level before diving into the data. I've added some annotations to the top to make this a little easier. The researchers varied whether the template was present as a control, as well as the nucleotides present, and the concentrations of CTP, UTP, and GTP. The big black line at the bottom is where the radio-labeled ATP ran to. The bands in the middle are abortive transcripts, and these variance bands at the top are full-length transcripts. Lane 1 is supposed to be a control, ensuring that we get no transcription products if we don't have a template present. We would expect to see no products other than this radio-labeled ATP. So what's with this product right about the tumor position? Turns out that in the synthesis of the radio-labeled ATP, a contaminating tumor was made that appears here. This tumor is the ATP plus an A at the end. We'll need to keep track of it because it's going to cause some strange effects down the road. In lane 2, synthesis should now be able to occur, as we've now provided the template and the nucleotide for the first two nucleotides in the sequence. We would expect to only see a tumor product to appear, as we don't have the rest of the nucleotides. But we see 3 and 4 more products here. What's going on? Remember that tumor contaminant causing problems in lane 1? It's at fault here too. Because it's a dinucleotide triphosphate, it can be incorporated in the synthesis reaction. As such, we can get more than the tumor we would expect, as it can be incorporated in place of any A. As such, every time A is added, we can add 1 or 2 nucleotides, resulting in these unexpected combinations. As a result, we can update our modeled results to include these options. In lane 3, we have now added all the nucleotides, yet in unequal stoichiometric ratios. This enhances abortive transcripts, as it is less likely that a required nucleotide after A will be present. increasing the possibility for an unstable short DNA-RNA hybrid to disassociate, resulting in aborted transcription. However, this still allows for the synthesis of full-length transcript. We see some full-length transcripts, but many more abortive transcripts, of various sizes up to about 6 nucleotides long. This makes sense given the model we discussed earlier. The hybrid is stable enough after making at least 6 nucleotides that even some hiccups in transcription, given the reduced concentration of nucleotides, will not result in the disassociation of the hybrid and the abortion of transcription. In lanes 4 through 7, we continue the conditions from lane 3 while increasing the concentrations of CTP, GTP, and UTP. We can see that the synthesis of abortive transcripts and the synthesis of full-length transcripts are inversely related, with more full-length transcripts being made as abortive transcripts decrease. However, even at equal concentrations of all of the nucleotides, as in lane 7, we still do see abortive transcripts, indicating that they still occur without enhancing for abortive conditions. Let's wrap up with some key takeaways. We can get enhanced aborted transcription by limiting nucleotide concentrations, resulting in RNA polymerase pausing more frequently and the short hybrid disassociating. We also know from adding the heparin that RNA polymerase is reinitiating without leaving the DNA molecule. This experiment also showed that we only get abortive transcripts up to about 6 nucleotides long. However, later work, such as the first experiment I showed at the beginning, actually showed that the maximum length of abortive transcripts is 10 nucleotides long. After about 10, the hybrid is stable enough that it can tolerate short pauses by the polymerase. However, the length of the abortive transcript is dependent on the transcription conditions and the DNA template. Lastly, even at equal stoichiometric conditions of nucleotides, abortive transcripts still occur. I hope this video was helpful. Thanks for watching, and go blue.